So for bio unit, section 1, we're focusing on microscopes and cell theory, right? So before people could actually invent, first of all, a magnifying glass, and even fancier magnifying glasses, which would be microscopes, we couldn't see things like germs. People thought spirits caused diseases, right? And they thought maggots and all kinds of other things could just grow out of thin air. That was called abiogenesis. There was a word where to go with the concept that if you left out a piece of meat, poof, there would be life coming out of it. Okay, now we know by paying attention that it was actually flies that came along and laid eggs and the meat grows. Right? So on your phone, there's an accessibility button where you can push magnifying glass. So we'll give that a try in class, I hope. So in your book, they're focusing on table salt. So this is zoomed in a little bit, like regular. This is zoomed in more, and this is zoomed in a lot. Okay, so what else do we got here? We've got the old school microscope. Okay, this guy Hook, Robert Hook, Bob. Okay, he made that, right? And it's possible that other people made them, but you got to write it down, right? People say, "Oh, I did my homework, but I lost it." Well, you don't get any credit for it. Same with these guys. So if you make it, you write down your observations, tell everybody, and then you win. Okay, you get to be famous for it. Okay, he made something about Micrographia with some pictures, right? You got to draw pictures of what you look at under the microscope. Okay, so cork, right? So there's a big thing. You're like, what's cork? The thing that you put in your wine bottle. Okay, so he cut little slices of that, and he thought, oh, the little boxes, which is the cell wall, right? Because cork is made out of a plant, and he thought, oh, those look like the little rooms that monks live in, like the religious guys. Right, or jail, technically jail cell. So it's like a cell compartment. So this info bit says, why did he call it that? Because it looked like little rooms. Okay. Apparently Aristotle took a look at some things too. Yeah. Okay. So here's a picture of his book, Micrographia. Oh, good. I'm glad there is a picture. Okay, so this is the cork. That's what cork looks like. I think it looks like a peanut. Okay, but if you slice cork really thin, it looks like this. And he thought that looked like little rooms. Sure, we'll go with that. Okay. Then we did a good job, because people always do, of improving on other people's designs. So Hook basically gets the credit for inventing it. And then this Anthony Van Leeuwenhoek. Okay, so that guy really took it to the next step, right? So when we're doing the models of the atom, there was Dalton, Thomson, Rutherford, Bohr, right? So each person knew the atomic model of the other guy, and then improved the design. Okay, so one of the focuses of this microscope stuff is learning about magnification. Okay, so these school magnifying glasses that we have, there's a couple of lenses. So you have the ocular lens, which is this tube, and that magnifies something by 10 times as big as it was before. Okay, then you have 3 objective lenses that you turn and they click. Okay, and one is a 4 times, and one is a 10 times, and one is a 40 times. Okay, they seem like kind of random numbers, but that's what they are. So it's a resolving magnification power. Okay, so 10 times 4 for the low power would be 40. 10 times 10 for the medium power, it would be 100 times as big. Okay, and then 40 times 10. The high power is 40 times as big as what it was. I find it's really hard to get them to focus though. So when I'm working with students, I basically never use the high power lens, and sometimes what happens is they bring the slide up too close and it actually cracks the glass. Yeah, fun fact. Okay, so in grade 10 you care about the field of view. So it's kind of easy. You just use a ruler, a clear see-through ruler, and you say, what is the field of view? Okay, and if they're really smart, they would slide this over to the 0, right? But in this case, you say, what is the field of view? How much can you see in this picture? Okay, and you might think, well, who cares? But we'd like to know how big something is. Okay, where people say, well, is a grain of salt bigger or smaller than a grain of sugar? Okay, so let's count. There's 1 centimeter, 2, 3, 4, and a bit. Okay, so I would say 4.5. If you're being really fussy, you might think, oh, well, there's a 0.5 millimeter here and 0.5 millimeter here, so that's 4.6. Sure. Okay, so that's 4.6 millimeters. And then a micrometer, this little upside down U, I think is called a mu, like a cat, mew. You just do some math. Okay, so 1 millimeter is 1,000 micrometers. So you take this number and times it by 1,000. Yep. Okay, so that's how you do that one. Cell theory, there's another video about that. Variables, we're going to focus on that with our celery experiment. Louis Pasteur, pasteurization, pasteurized milk. Okay, that's the thing. More about cell theory. This guy, Matthias Schleiden and Theodor Schwann, they came up with something. Okay, it has to do with cells. Ooh, pond water. We should definitely go find some pond water. I keep forgetting to do that. I'm going to write that down. Pond water. Pond water. Okay, and like I said, even with your phone, you can probably find some stuff. Okay, there's staining techniques. Okay, so a lot of things are clear, right? So it's kind of hard to see them when they're clear. So you use like beet juice or something that is going to make it so you can see what you're looking at. Okay, cells theory, definitely memorize those. All living things made of cells, all life functions take place in cells. They're the smallest unit of life. All cells produced from pre-existing cells. Okay, which for us now seems obvious, but it wasn't. Okay, these are stained and unstained cells, right? Clear, hard to see, stained, easier to see. Epithelial cells. For what? Oh, so these are skin cells, apparently. Nerve cells. What's this one? Muscle. Yeah, this is muscle cells. And, oh, fat cells. Excellent. Okay, nowadays, everybody knows what resolution is. Okay, because everybody has TV, right? And we have phones, and you know that if you zoom in too much on your phone, that it's blurry, right? And it gets pixelated like Minecraft. Okay, so even with security cameras, people have higher resolution security cameras, and I'm not too sure how the slang works. It's like 4K, you know, when you get caught for something, you go, oh, 4K. When I make sure I say it wrong, I say, hey, I 4K'd you. I caught you, I 4K'd you. Okay, so there's other kinds of microscopy. So that's a word for playing with microscopes. Okay, so we have bright field, dark field, phase contrast, differential, confocal microscopes. There's scanning electron microscopes, transmission electron microscopes, and this is something called a paramecium. That's like a little bug that makes you sick. Okay, what's this? A motor neuron, a brain neuron. So these are nerves. Okay, yeah, here's the words for the transmission electron microscope. So take notes on that, page 258. What else is there? Yeah, scanning electron, page 259. So you want to take a look at those. Okay, I'm sorry if you're getting dizzy here. Here's some other pictures of things under the microscope. So fun. Cell research is good for gene mapping. What else? Cell communication and X-ray crystallography. Okay, so go through and read this section of the book and take some sketch notes to go with your work. Okay, that's page 264. Okay, so we're going to switch from that to PowerPoint. Okay, and 3, 2, 1, boom. Okay, let's see how much we can get done in 5 minutes. So even if you can see really well, we can't see details as well as microscope can. Duh. What's this? blood cells. Okay, these are red blood cells, and I don't know what these ones are, probably white blood cells. Ew, eggs and sperm, yeah. Okay, so eggs are way bigger than sperm. That's what they look like under the microscope. Okay, these sperm cells have this little tail called a flagella that makes them spin. Okay, and that helps them move. Eggs don't have to move, sperm do. What's this? Looks like a piece of wood, but it's a human hair. What's this? A band-aid. Okay, so the fibers of the gauze are zoomed in. Hopefully not a used band-aid. Okay, what's this one? Don't know. You zoom in, oopsie. That's a flower petal apparently. So people basically look around and they think, what could I put under my microscope? Yeah, James Washington's telescope. Right, so a microscope was basically an upside down telescope. Here's some different designs. I've seen these ones in high school. I don't know those. This is the one that we usually use. Okay, and what you usually have to do in grade 8 is learn all the parts of a microscope, and the point of it is so that you know how to use it. Okay, so here's some history. If you put a little drop of water on some paper, don't put it on your textbook, but you could put it on something else, you'll notice it makes it bigger. Neat, huh? So these guys got credit for making the first microscope. Okay, this guy is the one we just talked about in the book. Okay, came up with a 3-lens system, good job, and he coined the name cells. There's this guy, Anton van Leeuwenhoek. He made it better. So we have a higher magnification, you can see smaller things, and he saw bacteria and called them animalcules. These are pretty. So the nucleus is in the middle and the cell walls are on the outside. Okay, so Robert Brown is like, I think there's a nucleus in the cells. They basically always use the letter E for this. The point of it is to realize that in a compound light microscope, when you're looking at something, it's upside down and backwards. Okay, and like I said earlier, your magnification is 40 under the low power, 100 under the medium power, and 400 in the high powered lens. Okay, there's a website we can click on. So here's your parts of your microscope. Okay, push pause and actually try to label it by yourself. Ready? Pause. 1, 2, 3, pause. Then I'll tell you the answers. Okay, so up top here you have an ocular lens, which is your eyepiece, so you look in there. And this part just kind of hangs on to the objective lenses. Okay, so you have low power objective, medium, and high. Then the part that you spin to get the different objective is a revolving nose piece. You carry it by the arm and the base. You should always use 2 hands. Not everybody does, but we should. Okay, this is the stage that you put the slide on, this black thing. Okay, then you have stage clips, which hold on your microscope slide. And you have a light source, right? You plug it in, and so you can actually see what you're looking at. And so there's a little lamp. And these 2 are for focusing. Okay, you will get yelled at if you use the coarse adjustment knob when you're under the high power, because it moves the stage up and down too much, and you can break your slide, which sometimes costs lots of money, like 25 bucks for one slide, if it's really cool, and it can smash if you're under the high power lens. So you just use the fine adjustment knob. So when you're focusing, start with the coarse adjustment, the big knob, okay, and then you refine it with the fine adjustment knob. Let's see what I can do in 1 minute here. Okay, yeah, so this is the part, this is the function. Take notes on that. Yeah. Oh, diaphragm I didn't mention. Okay, that's this little clicky thing on the bottom. And what you do there is they're different shaped holes in a little circle. And you spin that around to get different amounts of light that are let in. Your magnification I mentioned already. Low power, medium power, high power. Resolution has to do with your ability to focus. Okay, so this is, I don't know, some kind of plant, okay, because it's got square cells. So zoomed in a little bit more. Field of view we mentioned from the textbook. Okay, so how much can you